Hello and welcome to Fasting by Fungi podcast. I'm Dr. Gordon Walker. This is episode 14 where we're going to talk about fungal communication. And this is a really complicated topic, and to date there's still no real consensus about what communication is in fungi, how they do it, if there's any kind of sort of language per se. But they do have a lot of information, especially on the level of understanding yeast and the population dynamics of how single-celled fungi work. And we're still in the process of sort of like pulling apart and trying to understand how communication happens within mycelium of one organism, and then how it corresponds to mycelium of another organism. And there's a lot of really cool lessons we can learn from some of the model organisms that have been used as sort of the basis for science for the past 50 years or so. Saccharomyces cerevisiae is the classic yeast that was used for a lot of genetics, but we've learned a lot about fungi in general, particularly with regards to some of their secondary metabolites, how they respond to environmental conditions and things like that. And we're trying to translate some of those lessons to mycelium down in the soil, but it does live a fundamentally different lifestyle, although I think lessons you learn from fungi can often be broadly applied to the entire kingdom, as they all have similar ways of dealing with things, and all fungi tend to be kind of opportunistic, and so you see similar patterns repeated within that. So I'm going to just talk for a second about my life and what's been going on. It's Morel season here in California, and the season's been moving pretty quickly up in elevation. So about a week ago, I was finding stuff at like 3,500. I was up there recently and was finding it more like 38, 4,000. And things are getting absolutely plowed down by commercial foragers and enthusiasts. That's not necessarily a bad thing. It does make me a little nervous to see so many people out there hiking after like a big burn event, because ideally you'd want the morels to be able to disperse their spores. And so it kind of sucks that there's like people are out there at like 5 a.m. Commercial foragers are like pulling 30, 40 pounds a day. And it makes it hard as sort of an enthusiast who it takes me like 2+ hours to drive up there, and I can find maybe a pound or two. Maybe I need to find better spots that haven't already been picked over. But it is just exciting to be able to find some mushrooms, and I'm very hopeful because we're going to be getting a little bit of rain this week, and I'm hoping that'll slow things down, because otherwise the elevation goes up very quickly. If it's like 80, 90 degrees, the soil warms up and dries out before the morels even really have a chance to fruit. And like I said, I may do sort of an all morel episode soon, but I'm still like keying in myself to the habitat and trying to figure out exactly where to find them and what kinds of habitats are best for them. So it's exciting. It's morel season. I've been eating lots of good mushrooms, and I'm happy because after like a 3-month break where I really didn't do any foraging, I've been back at it. I've been working on my book, and I'm currently writing a chapter on edibility and the biochemistry of fungi, and that's been an interesting journey. And as I kind of go through the book and as I do this podcast, I'm learning lessons about what I need to include. And this week there was a publication that came out called "The Language of Fungi Derived from Their Electrical Spiking Activity." And while I didn't have an episode planned to talk about fungal communication, it seemed like an apropos time to address this, because especially if you look at the media surrounding this paper, people have read it. Journalists are not particularly good at reading primary literature and being critical of it. So all of the headlines about it are just very outlandish and make it sound like we've suddenly discovered this hidden fungal language, and that we're, a month away from understanding, being able to talk to mushrooms, which, spoiler, I don't think is really the case. And I'm going to, like I said, go over some of the sort of established hypotheses for how fungi communicate, and lead into things that seem they make sense to me, and then kind of end on talking a little bit about this electrical spiking activity and how that has been translated to a sort of quote unquote language. And I think all the work is really cool. I just am skeptical of calling electrical spiking activity in fungi language. So that's kind of where I'm going with what I'm going to be talking about. But I'm going to just dive into some of the different hypotheses for how fungi communicate. And some of these, again, are very well-established things that have been around for 40, 50, 60 years, things we've known about for a long time. And to that end, we've known that plants and fungi and other organisms all create electrical spiking activity. This is not new. This has been known for a long time. The desire to measure it and try to make sense of it has also been around for a long time. And the technology that's being used to do this isn't necessarily new either. I think it's just the people are taking the data and applying their own rationale to it, which can be kind of a dangerous place to be when it comes to data, because you should really let the data speak for itself and not have to do a bunch of manipulation to make the data say what you want it to say. Because as a scientist, I can tell you, quite frankly, you can always make data kind of go the direction you want it to if you process it enough, or you do enough stats or run the right algorithm. It'll kind of look the way you want it to, but it doesn't necessarily actually look that way. It just means you've massaged it to the point that it kind of fits what your hypothesis is. So talk about the hypotheses for how fungi communicate, right? So one of them is volatile aromas, and I'll give examples of these things, but volatile aromas are a thing that metabolites that are produced that come off, and fungi can sort of smell each other in a way. There are changes in pressure within hyphal cells, osmotic pressure gradients, and that's been theorized as a way of fungi communicating. There's sort of the physical disturbance, reshaping of the membrane through something called the cell wall integrity pathway, and that's also been fairly well shown to relate to fungal communication and the way that like different species can interact potentially, or even how fungi respond to physical disturbance in their environment. Small molecule diffusion, so secondary metabolites or nutrients that might be produced and are sort of diffusing across mycelium can definitely impact how fungi are communicating and responding. Cytoplasmic flow, sort of gradients, and a recent paper showed this really cool bi-directional flow, because it's been sort of theorized as, well, yeah, we know that like hyphae fungal cells are sort of elongated tubes, and they have what are called septa. So there's little holes, and so basically all the mycelium is essentially connected under the same sort of like cytoplasmic turgor force, and because of that, that's how they can swell so much with water, and they're so good at retaining and keeping water. And so the diffusion of molecules through hyphae has been, theorized as a way that they communicate, and that's been shown fairly well in science that you can have sort of small molecule diffusion. But the question remains, how do you coordinate fungal behavior across like a large network? Because armillaria, honey mushrooms in particular, can have very, very large, long networks. The biggest one is like a 3.5 mile diameter chunk of mycelium up in Oregon, the humongous fungus, Armillaria ostoyae, and I went up there last fall and visited this big chunk of mycelium. We didn't find any mushrooms because there was snow all over it, but we did see some mushrooms at lower elevation. We saw evidence of lots of rhizomorphs and other stuff, and that mushroom is able to coordinate decisions potentially across, or the mycelium is able to coordinate decisions across a very large, wide area. And the question remains, how you can't just do that through small molecule diffusion, because it's too slow, and you see fungi responding sometimes to stimuli faster than you would by just like this pure rate of diffusion across a mycelial network. And you see, mycorrhizal linking, and people talk a lot about, the mother tree, and Suzanne Simard has sort of done a lot of work to show that trees are able to send carbon and nutrients to each other via this mycorrhizal network. Is it communication, or is it really just like the movement of nutrients down a gradient, concentration gradient from high to low, or low to high? And that's sort of the idea of like a source sink thing, and that like maybe there's one really big healthy tree that's doing tons of photosynthesis and it's producing lots and lots of sugars. Those are being drawn down into the mycorrhizal network, and then they are dispersing to other nodes around the forest and getting distributed to different trees, but they're flowing out of like a very high gradient to lower gradients that potentially need it more. And that flow is not necessarily unidirectional. There could be bidirectional flow, where at other times of the year, maybe the flow of nutrients is going back into that big tree if the big tree is requiring more nutrients, like maybe in the spring when it's putting out leaves versus like the summer when it has full leaves. And this is a question of like, is that communication, or is it really just like flow of nutrients? And that requires us to look at what plants and fungi are doing and decide whether it's language. And I think that's a really important point to sort of recognize that like we shouldn't be too hasty to sort of over anthropomorphize what fungi and plants are doing, because they're fundamentally different than us as animals, and they don't have brains. They do with display intelligence, but it's a sort of this network intelligence, and it's not necessarily the way that we think of like language and communication as much as it is like a chemical. I don't know. I'm trying to think of a word that's not language. It's because it's not really a language. It's almost more of like a responsiveness kind of thing. You know. Anyhow, there's also, in terms of the hypotheses of way that fungi communicate, is this idea that electrical signaling could be used, and that's what this recent publication was about, because it's been sort of hypothesized that mycelium is the cells are sheathed with different like mannoproteins and beta glucans and chitin and things like that that could potentially allow almost like a action potential like thing to move down a mycelial strand, where the electrical potential would be jumping and you'd be like getting inrush of calcium ions. And there's a real temptation to say that mycelium underground mimics this sort of brain structure, because it is lots of interconnected things. There's nodes, but there are no axons, there are no neurons, there is no central processing center. For the most part, mycelium is fairly uniform in that doesn't have a whole lot of like specialized tissues the way that a brain would. There's not a whole bunch of different cell types necessarily. And I think scientists who are worth their salt would very much pause and say, hey, I know there's a temptation to say that like mushrooms are smart and that mycelium is like a brain or mycelium is like the internet, but don't say that. There may be good kind of quote unquote gateway concepts to help people who don't have a basis in science start to understand what's happening. But by saying mycelium is like the internet or saying that mycelium is like a brain, you're vastly overstating the science and what we actually do know. And I think in some ways you're denigrating what scientists are doing, because it is so much more complex and cool and nuanced. And if you say that we figured out fungal language or that, mycelium is a brain, people kind of tune out and say, oh, cool, we understand it. And they lose interest in a way because it seems simple to them. They can just apply what they know about brains, which is usually not much anyhow, to fungi. And it's so much deeper and more complex and interesting than just sort of this reductive, like, oh, mycelium is the internet and mushrooms are brains. They're not. And there's even the concept of mushrooms using light and bioluminescence to communicate, whether that's communicating with each other or that's communicating to insects. Again, hasn't been super well established. There's a hypothesis that bioluminescent mushrooms bioluminesce to attract insects at night to help them spread spores. And there's stuff to show that they can like spike their bioluminescence, particularly at night. So it's clearly something they're doing when there's low light. It's not just all the time. They're spiking that bioluminescence activity when it's dark. So they must be attracting something. But are mushrooms talking to each other with that? Or are they talking to insects? And I think that's a really cool thing to think about, like interspecies and interkingdom interactions, because we're thinking some ways about mushrooms and what their language might be internally. But we have to consider what is intracellular, like inside the cell communication, what is intercellular communication between two cells, and what is interkingdom communication between, or even like interspecies, different mushrooms, and then interkingdom when we have like mushrooms or mycelium interacting with bacteria or animals or plants. And so there's a lot of different forms this communication can take. And between all the hypotheses I just mentioned, what I really want to emphasize is that, like most things in science that are super hotly debated, these are probably all true. There's probably a situation or an organism or a thing where every single one of these hypotheses is absolutely correct and is the way that this organism might be doing some form of communication. Again, we have to sort of recognize like what is communication and what forms does that take. So I'm going to jump into some of the kind of classical fungal communication stuff and lessons from yeast. These are things I learned about when I was in graduate school. So there's a concept called quorum sensing. And quorum sensing is a way that like microbial populations will respond to the presence of a small molecule. And at a very low concentration, they might say, okay, we're gonna we're going to like turn our metabolism on in overdrive because there's a very low concentration of this molecule. But as that concentration of the molecule goes up because of their metabolism, they might sort of slowly start to ramp down their activity as that metabolite goes up. And that's a way of them saying, if there's lots of food, we want to be really active. But if we eat all of that food and we produce a bunch of this metabolite, we might want to slow our metabolism down. And like they're doing it by the presence of this quorum sensing molecule. So they're sort of adjusting their metabolism and their life strategy based on the concentration of particular molecules in their environment. And these are often, like I said, secondary metabolites or small sensing molecules, peptides, things that are produced. And it's a way, if you have a bunch of things in liquid culture, there's sort of a diffusion rate through this liquid culture, and that population is able to coordinate its activity in this culture based on the concentration of particular molecules. And that's a very classic thing. You see it in bacteria, you see it in fungi, you see it in lots of stuff. And it's a way for them to modulate their activity based on the availability of nutrients, on the environmental conditions, all this kind of stuff. An example of this in yeast, Saccharomyces cerevisiae, and fermentation is you start out with lots of sugar. Yeast say, hell yeah, we got lots of sugar. So they start going like crazy. They go exponential phase. As they're growing in exponential phase, they drop what's called the redox potential, which is sort of a chemical measurement of the amount of activity that can happen. And by dropping redox potential, they're sucking up oxygen, they're sucking up all the nutrients, and they are starting to deplete nutrients too. So when you get to really low redox potential and you have really low nitrogen, yeast metabolically start kicking out a lot of hydrogen sulfide. And hydrogen sulfide is kind of a stinky, farty egg smell that winemakers for the most part don't want. And so there's been efforts, some of my work was based on trying to like mitigate the production of H2S in fermentation. But what the yeast are using H2S for is as a quorum sensor, and they are coordinating their growth based on that amount of H2S that's present. So if you go to really low redox potential and a lot of H2S being produced, yeast take that as sort of an inflection point to say, okay, we're going to stop exponential growth and we're going to switch into stationary phase, which at this point they stop dividing and they continue to metabolize sugar, but they're no longer putting energy into producing more biomass. They're kind of just coasting, and they've recognized they're like, we've kind of run out of nitrogen. There's enough of us here at the party. We don't really need to make any more of ourselves. And this is not an all or none switch. This is sort of it's a gradient of like things that are responding to this H2S molecule. And over the period of a couple days, pretty much the whole population shifts from being exponential to stationary. And again, that's an important thing to recognize when we talk about quorum sensing and any form of communication, that it's not like a yes or no binary switch. It can be present in some tissue, or there can be a gradient of a response. And communication is very nuanced and complex, and that's part of why I don't like being reductive about fungi, because there is so much depth to this kind of stuff, and it's interesting to think about it. So another classic example of this kind of thing is Candida albicans, the classic like human pathogen, causes yeast infections and rashes and things like that. That will shift from a yeast phase or a single-celled yeast phase to like an invasive hyphal pathogenic form of being itself, based on the amount of farnesol and tyrosol, two sort of alcohols that are in solution. And that's related to nutrient status, environmental factors, pH, cell density, stuff like that. And so if we understand that, then we can potentially understand how Candida albicans, which is on our skin all the time, suddenly shifts to being like an infectious human pathogen. And so I think that's pretty cool to like get a good sense of like how quorum sensing is working in real time, and maybe how it's mediated by our environment. Like a rise in pH can cause Candida albicans to become more virulent, and that's probably because its metabolism is shifting, and maybe it's producing more tyrosol, so it goes into this like hyphal growth phase instead of being a single-celled yeast just on our skin. And that's there's spatial and diffusion limitation things that are interesting to think about as we think about like the microclimate that fungi particularly inhabits. And there's a really cool review I was looking at where they were trying to define sort of the Fungal Communicum, and this is like omics is like a fancy word for like trying to understand bigger picture things about all like the molecules, and the Fungal Communicum is like all of the molecules in different ways that fungi are working to process information and communicate with each other. Again, intracellularly, intercellularly, interspecies, and like interkingdom as well. And so that gets pretty complicated too, because there's a lot to potentially understand. The classic small peptide pheromone example of this is A and alpha factor, two small peptides that yeast make. And there's two mating types. It's basically sort of a fungal gender. There's A and alpha. And an A spore and an alpha spore will put out this small peptide, and one's saying, hey, I'm an A, and the other one says, hey, I'm an alpha. And if they have an A and an alpha get together, they form this cute little thing called a shmoo, where the circular spore creates a little like offshoot towards each other, and the offshoots link up and they share genetic information, and they become a diploid cell from two haploids becoming a diploid zygote, and then off to the races with being a yeast, and that creates a new colony. But there's other stuff, like the alcohols, the farnesol, the tyrosol I mentioned, and that can do like mediate morphological switching. You got fatty acids and lipids like oxylipins, and that can sort of mediate asexual to sexual switching. Volatile aroma compounds like the hydrogen sulfide I mentioned can mediate like growth, and when yeast make that decision to go from exponential to stationary. There's production of acetaldehyde, which can mediate glycolytic switching and sort of metabolic modes. So there's a lot of stuff. And a lot of this is like molecules are being perceived by potentially receptors in the membrane and are then affecting downstream cell signaling pathways like the MAPK kinase pathway, which is, I'm not going to explain this whole thing, but it's a very complicated sort of tiered system of cellular responses that spread out through the whole cell. And so like a single molecule being received by one receptor on the cell surface can then have a lot of downstream effects. And it's, again, a metered effect by like, if you only have like one molecule, it might be a little bit of an effect, but if you have a lot of this molecule hitting a lot of receptors, that signal is then amplified as more and more of this pathway, cell signaling cascade, is activated. And so again, that's where like quorum sensing and stuff like this comes into play. The way I like to think about nature and the way that fungi respond to things is a little bit like, I don't know if you guys have been inside of a music studio, but see those giant mixing boards with lots of little knobs and dials and all this kind of stuff. And I feel like internally, if you want to like try to understand what fungi are doing with all their different receptors and these cell signaling cascades, it's kind of like you go in and you're just carefully like switching and moving all those little knobs and things to like just get the sound just right. And as environmental factors change, it starts throwing off parts of your soundboard. So like some things get turned way up, other things get turned way down, and it changes the quality of the sound. It changes the metabolism, the lifestyle of what's going on. And there's so many different little inputs that all affect each other in that sense that it's really hard to pull apart sometimes just the activity of one thing. Because if you take one slider bar and just shove it up and down, you're not taking into account all of the other slider bars and knobs that have been like tweaked slightly as a function of a change, fundamental change in environment, like shifting temperature or shifting pH, which can have really wide-ranging effects on an organism, especially an organism that can't go anywhere. I think we as animals are always like, yeah, if it rains, I'll just take shelter. And like fungi don't have that opportunity. They sit in their food. They don't get to go anywhere. So what they have to do is retool their cellular membranes. And that's part of why we see so many responses that are sort of dependent on this like chemical perception stuff, because that's the only way they really can deal with stuff, is to restructure a membrane and understand the kinds of molecules that are in their environment. So there's also this cell wall integrity pathway, and what I'm getting at here with the cell wall integrity and like the MAPK stuff is this is how a lot of signals are perceived and then actuated through a cell. So, and again, we don't need to go into cell signaling right now, but just know that a fungal cell can perceive something in its environment and then make a bunch of changes based on sort of this idea of like a slider bar kind of thing and how much response is there. So, and the cell wall integrity pathway is one that is really about like intracellular communication, and it's about the extracellular matrix around a fungal cell, which is composed of the beta-glucans, the chitins, the manoproteins, and that's really sensitive to perturbations in the sort of physical deformation of the cell. So if you are putting it under osmosis pressure, or you're physically like pushing on the cell or pulling on the cell, it can change the programming of the cell based on the types of interactions that it's having. These are really sensitive to conditions and small molecules and perturbations of like, did the soil get mixed up? Did the temperature drop? Did someone like throw acid in the soil, something like that? Or did some organism create a situation that changed the fundamental chemical environment of where it was? And that's where important to understand too that like interkingdom stuff where a plant can elicit a fungi or a bacteria can like have a different thing, or a presence of another fungi can completely change the activity and sort of output of another organism, just depending on like how they're interacting and the molecules and the way they're perceiving each other in that environment based on some of the like small molecules and quorum sensing things and volatile chemicals that can go between these two organisms. But it really is like cell signaling cascades that then affect the metabolism, the morphology, the growth, the interaction. So this stuff is very complicated and isn't really what I would call language. It's definitely communication, but unless you're going to talk about this very diverse chemical language, and we don't think of lots of small molecules as being language necessarily. It's more just like the chemistry of the biochemistry of life, you know. So I think that's pretty cool. And again, I don't like being reductive about it and sort of saying, you know, they have a language, because I think it's the chemistry, biochemistry of life is so much more intimate and amazing, and you know, just more nuanced in some ways than language could ever be to some degree. Like language has lots of capacity for beauty and nuance and all these other things, but it fundamentally just functions differently than the way that small molecules in chemistry do. And so understanding that difference is important to understanding life. So I mentioned one of the most interesting things I read as I was sort of researching this podcast and trying to like get myself up to speed on fungal communication was a recent paper where they'd done some work. They're trying to understand the coordination of behavior across fungal networks in mycelium. And they were basically looking at a Coprinopsis species, I think it was Coprinopsis cinerea mycelium, and they introduced some nematodes that eat the mycelium. So it's a thing that hurts the mycelium. And what they saw was cytoplasmic flow of through specialized hyphal cells in the mycelium. So this wasn't like all of the mycelium was responding. It was like there's special microfluidic channels, specialized hyphae that were ultra small, that was having fairly quick movement of cytoplasmic components, so nutritional small molecule things like that, through these tubes running across the mycelial network. And it was moving pretty quickly. It was like 5 micrometers per second, which isn't incredibly fast. It's not as fast as an electrical signal, but it's pretty darn fast. And it was, the very cool thing they observed was there was bidirectional flow. So this is very similar to the idea of what slime molds do, where they do this cytoplasmic streaming. And cytoplasmic streaming is, in slime molds, is fairly dynamic. It happens pretty quick. It's like every like 90 seconds they'll switch flow of these intracellular components. And you can see that in my videos. I talked about it in the slime mold podcast I did, but you see the pulsing, sort of this breathing effect. And apparently mycelium can do this too. It just does it on a time scale. It's more like every like 2 or 3 hours it's switching bidirectional flow. But the important part there is that it's able to coordinate activity between like the leading growing tip and then like the internal parts of the mycelium, and it's able to like retool where it wants to go based on like where food is. It's able to maintain potentially memory, which has been shown by Lynn Body, that fungi mycelium can like remember where food was. If you kind of erase the path to it, they know how to get back to it. And it, I don't know, it's really cool, because here's like a mechanism by which we say, well, diffusion across the mycelial network isn't possibly fast enough for this for diffusion, chemical diffusion, to be a part of fungal communication. This says, actually, maybe there's specialized channels, like communication channels. Imagine those little tubes you stick like a message in, it goes whoop, and sucks it off to somewhere in the building. Maybe that sort of exists within the larger structure of mycelium, that there's these like specialized communication hyphal tubes that can quickly help coordinate activity at the surface of the growing tips and make decisions about that based with like the rest of what's happening in the mycelium. And it could spread alarms and say, hey, look, there's a predator, we should like retool. And you see those plants all the time. Plants get injured and they'll put off volatile molecules, and something else somewhere else will perceive that molecule and then know that it should like fold its leaves in because there's herbivores around, kind of thing. And so this was just a really cool experiment that was done. And it's made possible by the fact that like hyphae are those long tubular cells with a septa, and you can have fairly quick flow of cytoplasmic components, including nutrients, signaling molecules, and that's intracellular flow, but it's very responsive to its environment. And if you look it up, it's just Biodirectional Communication across fungal networks. It's pretty cool. That's a little review of one of the papers that did some of that work. I just thought that was really interesting. So I want to read you guys just a really quick passage from Merlin Sheldrake's Entangled Life, and then I'm going to jump into talking about the electrical activity. But this sort of starts talking about some of the original work that was done with electrical activity by a guy named Olsen, who I believe was a Norwegian scientist. But he had some Panellus stipicus, this bioluminescent fungi. He was trying to figure out how did it coordinate this pulsing activity, because they could observe pulses in the bioluminescence, and they put some electrodes in it, and we're trying to figure out like what was going on. So I'm going to just read this one little section. This is Living Labyrinths and page 61 in Entangled Life, if you want to go read this yourself. So Olsen chose the species of fungus carefully. He surmised that if electrical communication systems did exist in fungi, it would be easier to detect in species with a greater need for communication over long distances. Just to be safe, he chose a honey fungus, or Armillaria, the species that forms the record-holding mycelial networks that stretch over a kilometer and reach thousands of years in age. So that's what I was talking about, the humongous fungus that's up in Oregon. There's another big patch of it up in Michigan, too. When Olsen inserted the microelectrodes into Armillaria's hyphal strands, he detected regular action potentials, like impulses, firing at a rate very close to that of animal sensory neurons, around 4 impulses per second, which traveled along a hyphae at the speed of at least 0.5 millimeter per second, some 10 times faster than the fastest rate of fluid flow measured in fungal hypha. So that is true. This caught his attention, but in itself didn't suggest that the impulses formed the basis of a rapid signaling system. Electrical activity can only play a role in fungal communication if it is sensitive to stimulation. Olsen decided to measure the response of the fungus to blocks of wood, for which is the food of the species. Olsen set up a rig and placed a block of wood onto the mycelium several centimeters from the electrodes. What he found was extraordinary. When the wood came into contact with mycelium, the firing rate of the impulses doubled. When he removed the block of the wood, the firing rate returned to normal. To make sure that the fungi weren't responding to the weight of the wooden block, he placed an inedible plastic block of the same size and weight onto the mycelium. The fungus didn't respond. Olsen went on to test a range of other species of fungus, including a mycorrhizal fungus growing in the root system of plants, Pleurotus or oyster mushroom mycelium, and Serpula, the dry rot found fruiting in the oven. That's a different part of the book. They all generated action potential like impulses and were sensitive to a range of different stimuli. Olsen hypothesized that the electrical signaling was a realistic way for a wide variety of fungi to send messages about different parts of themselves. Message conveyed information about food, sources, injury, local conditions within the fungus, or the presence of other individuals around it. And so that is really cool work, and it's some of what Adamski has followed up on doing. Is he's essentially taking electrodes and sticking them in blocks of mycelium and looking at different pulses of the speed and frequency of electrical pulses and then the amplitude of that. And I think it's really fascinating stuff. It's very interesting science, but it's also really hard to interpret because we've known for a long time that plants and fungi and other things produce electrical impulses. And if you do something like put a flame to the mushroom or massively shift its environment, it probably shouldn't be that much of a surprise that you'd see a difference in these electrical signals. But the question is, what do they mean? And can we translate that in a way that makes sense, or are we just over anthropomorphizing what a fungus is doing without really paying attention to the basic biology and what we know about the biology and the chemical perceiving and the way that cell cycles and cell signaling work within these organisms? So Andrew Adamski is a pretty interesting guy. He works at the Unconventional Computing Lab, and he is a professor at the University of the West of England, Bristol, I believe. And he's done, I think, 3 papers now where he's sort of proposing this idea of a fungal language based on the frequency and amplitude of this electrical spiking activity that he's recording. And he's done previous things where he did it on pink oyster mushrooms, on Ganoderma, and in this paper, he worked on enoki, split gills, jack-o'-lanterns, and cordyceps. And basically what he did, so you take a block of mycelium, stick 8 electrodes into it, record a data point every second. He was doing like 78 point millivolts, 21 degrees Celsius, 80% humidity, and he did it all in the dark. So it was standardized experimental conditions, which is good. You want to see that. Read the methods of a paper to try to understand what this person physically did and how they recorded their data. And I think what it can definitively say is that these different kinds of mycelium show different patterns. And that's awesome. That's really fascinating. And I think that's really cool. But there's kind of a question of like, what does it mean, right? And this happens a lot in science. We're like, whoa, we have this cool data. And you're like, well, what does it mean? And people are like, well, I don't know. People do these biodiversity surveys of soil. And they're like, every time we go out, we find new organisms. We're like, well, cool. What does that mean? They're like, well, I don't know. It's sort of a thing. We're like, well, we're just doing it because we're doing it. And sometimes in science, this is called like a fishing experiment, where you're just like, well, we just did something to do something because we wanted to see what would be there. And that's really hard to get funding for stuff like that, because unless you have like a clear defined hypothesis that you're actually working towards, most places won't grant you money to just like do what you want to do. Which is why sometimes stuff like this falls to the cracks or falls to particular individuals who are able to fund their work or do their work outside of an academic sphere, or even just in the academic sphere, but kind of like, this is fairly low-tech work to some degree. Like, he's just kind of sticking electrodes in a block and recording pulses. You could literally do this at home, most likely, if you set the conditions. The cool thing he's doing is he's applying a lot of math to the electrical impulses, and he's filtering the data to try to remove some of the noise. So he's looking at like outliers from the data set and saying, well, nothing over this amplitude, nothing under this amplitude, nothing that's like too frequent. So he's trying to remove some of like the electrical noise that happens when you record data like this, which I think is a good idea. But you are, again, biasing the data set by removing data points that could potentially be relevant. And the thing that kind of struck me as a little bit disingenuous or a little snake oil about this is he has a line in this paper where he claims that fungal language exceeds European language in morphological complexity. And then later in the paper, he basically sort of says one species he looked at had like quote unquote 50 words, which I'm not totally sure how you're supposed to like say that fungal language has more complexity than human languages if there's only 50 words. So that didn't totally sit with me, and maybe I'm just misunderstanding the work that he did. But if you read this paper very early on in the abstract, it says, assuming the spikes of electrical activity are used by fungi to communicate. And so his entire assumption on this paper, it's an assumption, right? It's not that he let the data speak for itself and he said, well, this is definitely fungi are doing this because of this. He's just, we're assuming this, and then I'm going to do a bunch of math to fit the output of the data that I want. And to me, that is not conclusive proof that there's language in fungi. And I think it's kind of a dangerous thing to over anthropomorphize what's happening here. Not dangerous, but it's just like not real. I think unless we're really linking the stimulus response to cell cycle changes, to cell signaling, to changes in metabolism, what we're looking at with these blocks is generally just kind of internal chatter, maybe, or just like electrical firing and signaling stuff that could be based actually on real quorum sensing or other forms of communication that we better understand in these fungi. And that's where I'd like to see the link made between like what do these electrical signals mean as they relate to metabolism, to nutrient cycling, to other things that are a little bit easier to understand in terms of like what is communication on this chemical level. And because I don't think that the science of this electricity and spikes in electrical activity in fungi is going to be separate from the rest of the fungi. I don't think that there's like a consciousness and a language that they're using that's somehow separate from all this other chemical chatter that's happening as well. And, again, I'm skeptical of anything where you use a lot of like math and algorithms to kind of dictate language. And again, I'm not a linguist and I don't have a great statistical math background, so I didn't totally understand everything he did, but it did seem a little bit like a reach that he was trying to say that because we've seen different patterns in electrical activity, that somehow there's a fungal language. And I think the hard part is that journalists read this and put out these sensationalist headlines because it sounds really cool to say, oh, we figured out fungal language, and really notable mushroom people had then parroted the same thing and say, look, we figured this out. And like, well, pump the brakes a little bit. Let's maintain some skepticism. Let's maintain just some sense that like this is super fascinating and I'd love to see more follow-up work, but I'd also really like to see deeper work done with like getting into omics, looking at metabolomics, looking at transcriptomics, looking at the changes that are physically happening within a cell as it relates to these spikes in electrical activity. And I think like there is conclusive proof that if you grow fungi in an electric field, it looks a little different than it would have otherwise, and that's not surprising that like many proteins in our animal cells, if you grew us in an electric field, we wouldn't be normal. Right, most life, if you grow in an electric field, is going to be kind of weird because we have plenty of like proteins in our bodies and cells that can respond to electrical fields. And he did do some in a previous paper. He'd done some work with like pink oyster mushrooms where he'd like put a flame to it, and he saw a big spike in the frequency of electrical activity. So he was sort of like linking stimuli and the electric response, but it wasn't really clear what the output of that was. So it's like clear you can link stimuli and the response, but then like what's the output of that response? And that's where like it's not really a language until you have those like three pieces communicating with each other. And so I guess my takeaway from this, the presence of electrical activity alone does not imply quote unquote language, and you haven't really shown that the electrical impulses can link stimulus to a response in a meaningful, actionable way. So I don't know. I'm skeptical. I think it's really cool. I'd love to see more from this. I'd love to talk to Adam. Or Andrew. He's a pretty interesting guy. But I'm skeptical that we've really figured out like fungal language. And like I want to end too because there's another thing that goes viral constantly, and this is something that really bugs me. Is there's all these sort of like plant wave things. It's like a little set of electrodes you can clip onto a plant or a mushroom, and they make noise. And people are like, oh my god, the mushroom's making noise. The mushroom's singing. The mushroom's doing this. And you're like, nope. That's just a synthesizer with a set of sounds that someone has programmed into it, and it's responding to those electrical impulses, is responding to the frequency and the amplitude of those electrical signals. But the output, the sounds that you hear in those videos, is something that somebody has programmed in there. You can program in heavy metal, you can program in weird alien sounds, you can program in whatever you want. But it's not the real noise that the organism is making. And it works just fine on plants, it works on mushrooms, it works on really any living thing that produces these electrical activity. And that is not proof of mushroom language or that mushrooms are somehow have a vibe or anything like that. And I hate to say that because I know people really like to believe that, and it's like very compelling that, oh, look, mushroom music. It's all BS. And I have some friends who do it, and it's really cool. I like it. It's interesting. And I think as long as you're honest about what you're doing and how it's done, it's totally fine. I don't see any harm in it, but I do see a lot of people kind of hooking it up, and then maybe they don't understand the science well enough to explain it, or maybe they're just like intentionally being misleading and be like, yeah, mushrooms make music. And then they're like making money on Spotify off of this, just like random like noises, or whatever music they program in, whatever, it's fine. You gotta respect people's hustle. But I will say that way of making noise is kind of a contrived thing, and it's not real. And I think that, maybe, like I said, about being cautious about over anthropomorphizing mushrooms is that we need to always go back to the science and understand what we've learned as the basis, and think about scientific consensus often as like a bell curve, and you want to look for consensus that's within the bell curve, because if you have these little outliers, doesn't mean that your model's wrong, because these outliers don't fit. It may mean you need to shift your model. And like I was saying, I think of all the forms of communication that we've understood about fungi, this electrical activity is something we clearly don't understand its purpose. And I'd really like to understand its purpose in a much deeper, potentially more scientific way before we just start doing math to try to like invent language. And they've done this, I know, with like whales and stuff like that, and they are able to see with whales and stuff there are some patterns and ways that they speak to each other, but whales are very different, right? They have brains, they're mammals, they have capacity for language and memory and thought in a way that like mycelium doesn't. And so let's be careful to not kind of project our feelings and our thoughts onto mycelium, just because it's an attractive concept. So anyhow, that's my my brain dump on this stuff. I think it's super fascinating. I'd love to learn more about it. I'm really excited to read more in my time about fungal communication. But I do think that the one that I learned about that I thought was really cool was those sort of specialized hyphae that send nutrients in a biodirectional way much faster. And to me, that seems like a pretty good explanation for how a lot of behavior is coordinated across fungal networks and within mycelium. And the thing that I'm really fascinated about that I need to do a lot more reading on, and I'll probably go back to some of Suzanne Simard's and Daniel Durrell's publications, is how different mycelium from different species interact, and then how like nutrient transfer and carbon and nitrogen flow through those mycorrhizal networks, linking together trees in the mycorrhizal sort of hollow forest organism that is the woods. Anyhow, as always, if you guys want to learn more from me, you can check out fastingbyfungi.com. I've got cool merch and t-shirts on there. I've got an About Me page. I've got, what else am I doing? Oh, I've got a mushroom discount page. You get discounts on mushroom kits. I've got a Patreon, so if you want to support me, you can go there. Please follow me on YouTube, on TikTok, Instagram, Facebook, Pinterest, Twitter, all those things. I'm working on my book. I'll be posting updates about the book on Patreon. And I'm just excited, as I got a few other podcast interviews I did recently, and hopefully those will be out soon, so I'll share them with you guys. And yeah, wishing everyone a wonderful week, and much love to you all. So bye-bye.